So welcome to the video on Chapter 13 on controlling microbial growth. This will cover sections 13.1, point 2, and point 3. So let's start with 13.1, the control of microbial growth. For this chapter, I'd like you to know certain definitions such as disinfectants, antiseptics, and sterilants. I'll ask you to describe the principles of controlling the presence of microorganisms through these various techniques. So basically, why do we even want to control their growth? I'll ask you to be able to differentiate between microorganisms of various biological safety levels. There'll be biological safety levels 1, 2, 3, and 4, and to explain the methods that are required in handling these microbes in the lab. So let's start with the first question. Basically, what is it meant to be clean? So how clean is clean? And what I'm hoping that you'll know is that the answer will really depend on what that item is being used for. So in this case, that item is called a fomite. A fomite is anything that's inanimate, non-living. So inanimate objects such as the doorknobs, your cell phone, toys, towels, anything that is not an organism and that is going to have microbes all over it. Something that you could use to actually spread disease. So to help answer how clean is clean, here's a dishwasher. Now, would you, after going through the cleaning cycle, would you say that these dishes are clean? And the answer I would think that you'd give is yes. They are clean for what they're being used for. They're being used to serve food on. However, they're not clean enough for surgical instruments to be used in an operating room. So yes, they are clean, but not clean enough. And these dishes are a good example of a fomite. So we need to think of how clean is clean and how to make these objects clean enough for their intended use. So for instance, these surgical instruments would need to be completely sterile because there's still some microbes present on those plates and in those glasses. So here are certain reasons of why we want to do this. In order to clean these, in order to have clean instruments or clean fomites, they're going to prevent or reduce the control of microbial growth. We want to bring down that amount of growth down, if not 100% zero, to at least down to 10% or something around there. So the first reason that I'm giving you of why we want to reduce the amount of microbial growth is one, to protect yourself from being infected. Again, if you were to eat off dirty dishes that hung around, these microbes would grow in the food, they would increase in numbers, and you'd increase your chance of getting infected. If you put those dishes through a dishwasher and you eat off clean dishes, you've reduced the chances of becoming infected through these dishes. Another reason is to prevent contamination of the microbial culture. This is more in the lab. And so you've kind of practiced this in the labs where you've sterilized your loop by flaming it and then touching a colony and then spreading it onto other media, whether it's a slide or another biological media. You're sterilizing that loop so that you don't contaminate the microbial culture that's present on that plate. This is also done in clinical labs because if we're going to identify the causative agent that the patient has that's causing the disease in the patient, we need to start out with a culture that's pure. We don't want to introduce something else into the sample. The third reason is to prevent spread of human disease. So if you don't contaminate yourself, you're not going to contaminate others, as an example. Okay, so there's two terms you're going to need to know. First one will be sterilization. What does it mean to be sterile? So sterilization is the complete removal. I'm stressing the word complete. The complete removal or the killing of all vegetative cells. So they might still be present and might not be removed, but they're completely killed. Also including, the vegetative cells include the protozoas, algae, bacterial cells. Remember, bacteria make endospores, and endospores are the most hardiest dormant stage of any microbe. So you have to also either remove or kill endospores. And viruses, even though they're not living, you need to destroy them so that they cannot enter cells. So whatever you're sterilizing, whether it's liquid or a solid surface, you have to remove all the microorganisms, or you have to kill all of them so that they cannot somehow replicate. Any chemical or any, basically it is just chemicals, any chemicals that are used to sterilize an object, they're called sterilants. So the definition of a sterilant: chemicals used to kill or destroy all microbes. Compare sterilization with aseptic techniques. Aseptic techniques are protocols or methods used to prevent the contamination of sterile surfaces. So you're going to start out with a sterile object. For instance, you're handling surgical instruments that should be sterile. That's what these instruments will be clean enough, only if they are sterile. Now, you as a physician or an OR nurse helping out, you can't just touch those instruments. You have to use aseptic techniques to prevent the contamination of these instruments so that the patient doesn't get infected due to the operation. So these are protocols. These are steps that are used to prevent the spread of contamination. I'm throwing this picture out there. This is a picture of an autoclave. This is typically how we sterilize a lot of solid objects. This is used by heat and pressure. So basically it brings the temperature of steam higher than boiling temperature, up to 121 degrees Celsius for approximately 30 minutes. Here's a cartoon just showing you the steam that goes in that is surrounding the objects. Remember, water is steam. Water is a good heat inductor or conductor, so that it's going to make sure that the chamber inside this autoclave reaches 121 degrees Celsius. And it's pretty normal to do this for 20 minutes. I'll come back to heat sterilization later in this slide or in this presentation. And again, just a quick note to say that autoclaves are perfect for these items here on the right, all these solid metal operating utensils. However, you have these plastic medical devices on the left. Heat sterilization might not be the best because you might damage them. So we'll talk about another method. Bring back my first talk on the cell. I talked to you about Lister, Robert Lister. Joseph Lister, and he introduced carbolic acid. So he had this spray where he sprayed his hands, the instruments, the bedding, etc., to try to sterilize the operating room as much as he could. He didn't quite understand what he was doing at the time, but he did reduce the spread of infection to his patients during operation. Today, you look on the left, we use ultraviolet light. So this is a robot type of device that basically will UV irradiate the entire room. And what UV rays do is they damage the DNA. So if there's any cells present, including your cells, where the UV light can penetrate, it will go through the cells and enter the chromosomes. And basically, if there are two thymine nucleotides together, we call it a thymine dimer, it will covalently link them, thus damaging the DNA, preventing DNA replication. So in this case, UV radiation doesn't sterilize, but completely removing the organisms from the operating room, but it will completely kill the organisms that are there. So this is used for sterilization. Just to remind you, what you've been practicing in the lab are aseptic techniques. Here's a lab technician flaming a loop before placing it either into a broth culture or transferring that broth culture to a solid media. And either way, before and after using these loops, you want to flame sterilize them. Again, aseptic techniques, it helps prevent contamination of those microbes. I told you that you want to protect yourself, and in the lab we use personal protective equipment, also known as PPE. In this case, you would use gloves, a lab coat, a hairnet if necessary. It all depends on what level, and possibly a mask or eyewear. but definitely gloves and a lab coat. And this is, again, to protect yourself, and where they're filling up medical devices or medical solutions, they also want to make sure that the medical solutions stay sterile. So they're using aseptic techniques to prevent the spread of microbes and prevent the contamination of these medical solutions. Again, here's a person working on a microbiological sample, and they're using aseptic techniques to protect themselves and the solution. And we can't get away with aseptic techniques if we don't talk about hand washing. It is the best way of preventing the spread of disease. These days, if you go to a dentist or a dental hygienist, they will also wear personal protective equipment. The only problem with this picture here is that this dental hygienist or dentist did not button up the front of their lab coat. Otherwise, they're using gloves, they have a mask, they have a shield protecting their eyes. They just need to button up that lab coat. Again, they're protecting themselves, and from one patient to another, they would then change the mask. They would definitely change gloves. They don't always change their lab coats, but nonetheless, it's the gloves that come in contact with the patients. Okay, so medical procedures with a risk for contamination should be carried out in a sterile field. So it's not just the physician or the personnel in the operating room that should be covered, but as I mentioned, that the operating room should also be UV irradiated so that the entire room is a sterile field. The only object in there that would be considered non-sterile is the patient. Okay, sterilization versus disinfection. So remember, sterilization was the complete removal or killing of all the microorganisms present on that fomite. Disinfection is a procedure to inactivate most, not necessarily all, but most microbes on the surface of that fomite using antimicrobial chemicals or heat. So some microbes will remain, thus the fomites are not truly sterile, and endospores in particular are not killed by disinfectants. Okay, so disinfectants should, as a guideline, when they're being approved by the FDA, should be fast acting. They should be stable during that time of being used, easy to make, very inexpensive, and easy to use. Now, I'm not going to stress these features of what a disinfectant should be, but I'd like you to know an example. And I'm trying to use common household examples. So an example of a disinfectant would be chlorine bleach or any type of chlorine products. The way that they work is they basically will oxidize molecules in the cell, which will help kill them. So we already know that oxidization breaks down phospholipids or glycolipids. It separates the glycol molecule, the glycerol molecule, from the fatty acids. Therefore, the cells will fall apart. And it also denatures proteins. So by oxidizing microbes, you will kill the majority of the microbes present. This is a picture of disinfectant wipes that I have at the house. It claims to kill 99.999%. Sounds very good, of bacteria. It kills cold and flu viruses. It specifically mentions staph. Everybody's worried about staph and MRSA, or MRSA. Kills E. coli, salmonella, strep, all the good ones that most people know. And the ingredients, I'm not sure if I can pronounce it, but it will be the dimethyl benzyl ammonium chloride. There are others, the ethyl benzyl ammonium chloride. Again, they are chloride molecules. That's the important part. And again, they disrupt the cell membrane by oxidization. Where would you use these disinfectants? Again, on fomites. So in the clinical setting, they're very important in the examining room, or even the patient's room when they clean the floors, they clean the furniture. Examination rooms, operating rooms. Operating rooms would have to be cleaned before sterilized, but again, operating rooms would have to go that one extra step of sterilization. Okay, so antisepsis. Antisepsis is a procedure for killing microbes with antiseptics that are safe for use on tissue. So you're going to distinguish between disinfectants and antiseptics. Disinfectants, remember, are to be used on fomites, on inanimate objects. Antiseptics are going to be used on people or patients. So they have to be safe to use on tissue. These antiseptics are antimicrobial chemicals that are safe to be used on living skin. Again, they are not sterilants. So they will disinfect, but they are safe to use on patients. They have to penetrate the tissue without causing major damage to tissue. Again, nothing is 100%. If you leave it an antiseptic long enough or in high concentrations on a patient, you could cause irritation or burns, but if used correctly, they will not cause damage to that patient. And two such examples that you probably have at home or that you probably use at your job is hydrogen peroxide and isopropyl alcohol. So here's hydrogen peroxide on the left, and if you remember when we talked about aerobic cellular respiration, our cells, as well as any microbes that are able to undergo aerobic cellular respiration, do so because they have the enzyme catalase that destroys hydrogen peroxide that is formed at the end of the electron transport chain. Well, if you supersaturate these cells with hydrogen peroxide, you will then oxidize the lipids and the proteins and thus destroy the cells. And that's what you're doing when you're putting this on a cut. It's an antiseptic, which is cleaning a wound, and it's going to kill the majority of microbes that may be present in that wound. Likewise, alcohol. Remember, you've used 95% alcohol to dissolve the cell membrane and the outer membrane of your bacteria when you were performing gram stains. So you used 95% alcohol. In that case, it was ethanol. In this case, this is isopropyl alcohol. But in both cases, it denatures proteins and, more importantly, dissolves those lipid membranes, thus, again, disrupting the cells. Okay, so in conclusion of what I've presented so far, you should know about sterilization. It's not present in this slide, but sterilization removes all the microbes 100%. Compare that to disinfectants and antiseptics, which only remove the potential pathogens, so the majority of microbes, and if you get the majority of them, then you're more likely to get the pathogenic microbes that could be present. Compare or contrast the disinfectants with that of antiseptics. Disinfectants are only used on inanimate objects called fomites. Antiseptics are safe to use on tissue. So in both cases, the microbial load is reduced, but sterilization is not achieved. Okay, so now let's move on to the next objective, and that is to discuss biological lab safeties. So laboratory biological safety levels. These are measures that are put in place to prevent the risk of lab workers from becoming infected with certain pathogens. So it's there to protect the person working in the lab. Now, it could just be, it's not just lab technicians, it could be any healthcare provider that will come in contact with a person who's infected with these microbes. So some microbes are more easily spread than others. Some cause more lethal diseases than others. Some don't cause any harm at all. So the NIH, National Institute of Health, the Center for Disease Control came along with guidelines and basically categorized all microbes under 4 laboratory biological safety levels. We call them BSL 1, 2, 3, and 4. BSL 1 being the most mild organism, and BSL 4 being the worst or the most pathogenic, the one that's easily acquired and that could often lead to death. So we're going to start with the BSL level number 1. Okay, so I've mentioned quickly that microbes are classified to these 4 groups, and these are the 4 criteria that were used to classify these organisms. 1, are they easily transmissible? Now, the easiest way to transmit any microbe is through aerosols, by breathing it in or taking it in through your nose or your mouth. probably the most difficult one is if you had to inject it through your skin. So, these are considerations. Now, how easy is it to infect humans? Now, some microbes don't infect humans at all. They may infect our pets, or they may infect plants, but they don't infect humans. If they do infect humans, what disease do they cause? Is it just a mild illness, or is it death within 3 minutes, as an example? And type of work that's done on the microbe, that depends on the situation. Also, that's probably not here is the next one is, well, is there a treatment? Are there any vaccines or any drugs that once we know that the person has this disease, can we treat them to prevent death from happening? So let's start with BSL-1. So I have here a definition, this comes from your textbook, that these microbes generally do not cause infection in healthy individuals. And the key word here is healthy human adults. So healthy. So you have to have a strong immune system. Now, if you are immunocompromised for any reason, well, you may become infected and may have a disease through an organism that is classified as a BSL-1 microorganism. But otherwise, a normal, healthy individual does not get infected under normal means by a BSL-1 organism. So in other words, they pose very little risk and very few precautions are needed. Now, while working in the lab, what you need to do is work on an open lab bench, or you can work in an open lab bench without containment equipment, similar to your classroom. You'd need a personal protective equipment such as a lab coat, gloves, and eye protection. Now, we only provide eye protection in the lab if we feel that there is any risk of splashing any liquids in your eye. You're asked to wash your hands before and after working with these microbes. And eating and drinking are not allowed at all in that laboratory space. And any potential contaminants must be decontaminated before disposal. So any items that came in contact with those microbes should be placed in a biohazard bag, and that biohazard bag must be properly handled before it leaves the laboratory. The lab doors have to separate the lab from the rest of the building. Now, this sounds very similar to your classroom. In fact, our classroom is, we could use a BSL-2 organisms in our classroom, but we choose to only work with BSL-1 microorganisms while teaching microbiology at Massasoit. Okay, let's go to the next level, BSL-2. These microbes pose a moderate risk. So let's go back to the previous slide. Remember, BSL-1 generally are not known to cause disease. Well, BSL-2, they are sometimes related with moderate risks of disease, especially with the person handling it. So in this case, we would be in the lab. These microbes would include those that cause a very mild disease, or those that are very difficult to contract through aerosols. So generally you would not get them by breathing. You may get them by an accidental injection or cut in your hands. That's why it's always good to wear a Band-Aid and a glove on top of those Band-Aids. So although they do pose a moderate risk, under the conditions of using them, that risk is actually limited or very minimal. So what precautions do you need? You'd need the same ones that you would use while working with a BSL-1 microorganism. But in addition to those precautions, you would have on top of that some training before you could handle these microbes. You would need to wear a face shield. So whereas before you might have eyewear, some goggles, in this case you'd have to go one step further to protect your full face, not just your eyes. So you'd need an eye shield. And you'd most likely work under a biological safety cabinet, especially if there would be some splashing involved during the procedure. These organisms absolutely have to be autoclaved before they leave the lab. That means autoclave is sterilization. Remember, I showed you a picture of an autoclave. And you have to have an eye wash station immediately available in case you do get splashed in your eye. And then this must be followed up with a visit to your primary care physician. Now again, here's a biological safety cabinet. So it is a It's a negative pressure type air in which air is sucked into the cabinet. A technician would be working on the outside of that cabinet where their hands only exposed inside. The airflow would prevent any microbes from coming out or coming in. There's a HEPA filter inside that cabinet, so therefore you would prevent any contamination through aerosols. The aerosols would be lifted through that air current and enter the HEPA filter. And on the right is an example of a face shield that could be used, and here's a technician actually working inside a biological safety hood. She's pipetting some microbial cultures, and at the end of the day, this cabinet is completely wiped down and UV irradiated to sterilize the inside of that cabinet. So let's go to the third level, the biosafety level 3 microorganisms. Now these don't pose just a moderate risk. These are basically, they do cause lethal infections if you inhale them. So, the potential is very strong that once you inhale it, you will have disease. It's not just moderate, and it could be lethal. So an example is Bacillus anthracis or HIV. It does require extra precautions beyond that of BSL 1 and 2. And these personnel are under medical surveillance. So you may have to have your blood drawn occasionally to see if you've been exposed. You'd have to be offered immunizations if there's a vaccine to help reduce any risk of possible infection. Your PPE or your personal protective equipment would have to include a respirator, so a face shield is no longer sufficient, and you'd need to have a full face mask that has a respirator attached to it. All the work has to be done, not just some of the work because of splashing, but all the work must be done under a biological safety cabinet. And the personnel must wear a solid front protective clothing. So your lab coat is not solid front because it buttons in the front. These gowns would have to tie in the back to prevent any microbes from passing through the your lab coat and entering to your clothes. Now, when you're done, these gowns have to be removed before leaving the lab, and they must be decontaminated. And the lab coats must be constructed to promote containment. So they can't be made out of just simple cloth material because cloth absorbs liquids and it could seep into your clothes underneath. There's 2 sets of closing doors. Actually, the lab coats, but the labs must be constructed to promote containment, meaning that if there are any microbes present in that room, they should never leave that room. There'll be 2 sets of self-closing doors, and there'll be some negative air pressure. And again, the lab coats cannot leave that room. And normally these labs are locked, and they'll have a digital code that only personnel that are allowed in that room has access. Also, there'll be a directional airflow, so there'll be negative pressure so that the air comes in the room as opposed to air exiting the room. So the air coming in the room will be clean air, and of course you have to remove that extra air, and the air as it leaves the lab will be filtered through HEPA filters. So here's a picture of a technician that's working under a biological safety cabinet, and he has a respirator attached to him, and his lab coat does button in the back. So he's equipped to work with a BSL-3 microorganism. The most deadly organisms, an example here is the Ebola virus, that will be the most dangerous and often fatal microorganisms. So you don't want to have any chances of getting exposed to these microbes. They're usually easily transmitted by inhalation, and there's often no treatments or vaccines at the time. So again, extra precautions far and beyond the BSL-3. You must change your clothes before entering, and you need to shower after, upon exiting the lab. All materials have to be contaminated, so whatever you brought in with you has to be contaminated before you can take it out. You have to have a full body protective suit with an air supply attached to it, and you may also need a biological safety cabinet. All the air that's leaving the lab has to pass through 2 HEPA filters, not just 1. And the lab has to be completely isolated from the rest of the labs. So these are just pictures of the personal protective equipment needed for the different biosafety levels, 1, 2, 3, and 4. Again, the major one are the face. You go from goggles to a full face shield to a respirator to a full body suit, basically. Okay, so the learning objectives for this part of the lecture was to compare disinfectants and antiseptics and sterilants, describe the principles of containing the presence of microorganisms, and to be able to differentiate between the different levels of biosafety level microorganisms, and explain the methods that you would use if you were to handle these microbes. And always be aware, today in the lab, in the classroom, or in the future in your job, what safety levels do you need to follow when handling certain microbes or when examining patients. So again, it's a way to protect yourself. So let's go on to the next section, that's 13.2, and we're going to describe the physical methods used to control microorganisms. Okay, there's going to be different physical methods. The common ones will include high temperature as well as low temperature. So temperature is big. Radiation, either with gamma rays or UV rays. I won't discuss filtration, but filtration is basically filtering liquids through a certain pore size membrane. And if you use a pore size membrane that is smaller than that of the microbe, then you can separate the microbe from liquids. So filtering liquids is a very good way to sterilize certain liquids. And then desiccation, drying out, and other methods. So many of these methods they will non-specifically kill cells. So if you treat some of your cells, it will not differentiate between your cells or microbe cells. It will disrupt their membranes. It will change their membrane permeability to allow a lot of water in or liquid out. It will damage their proteins and their nucleic acids by denaturing, so they will not be able to replicate or metabolize. And they'll probably do some chemical modifications to their DNA. So either way, these organisms will die and will not be able to replicate. So let's start with the most common method, and that is heat. So heat is widely used. It's been used for many, many years. When we talked about spontaneous generation, we talked about John Needham, Francisco Redi, Louis Pasteur. They all heated some flasks with broth, and they basically would have sterilized this broth. So heat's been known for a long time as an effective method of controlling microbial growth. And if you do heat these organisms long enough, or these broths long enough, these liquids, you will sterilize them. Okay, so there's a couple terms you need to know. The first one is thermal death point. It's important to know that for each of the objects that you're sterilizing by heat, and that's at the lowest temperature that you need to obtain before all the microbes are killed within a 10-minute exposure. So the cutoff is 10 minutes. So at what temperature will you need to reach in order to kill all the microbes at 10 minutes? That's the thermal death point. The thermal death time is the length of time needed to kill all the microorganisms in a sample at a given temperature. So you can try different temperatures and see at what time it takes to kill all these organisms. So for instance, if the thermal death point in the previous slide was to measure the amount of the temperature used to kill all the organisms at 10 minutes, we didn't achieve it with this temperature, because at this temperature it took a little bit more than 22 minutes. So we would have to raise the temperature based on this graph until this graph actually intersected at time 10 minutes. So the thermal death time would be experimented at different temperatures to see how long that each temperature takes to kill all the organisms. And the very last temperature, the temperature that kills within 10 minutes, would be defined as the thermal death point. Okay, so we're talking about, basically with that broth, we're talking about liquid or moist heat sterilization. That's the common type of heat sterilization, but sometimes we need dry heat sterilization. So dry heat sterilization is an example of what you've been doing in the lab. So you've been flaming your loop. Some labs don't have a Bunsen burner, so they use this incinerator and the right, in which it uses electricity to heat up a coil. You stick your loop in that hole, and basically it will achieve the same result where it will heat the loop. So it's a way of doing it without gas. So dry heat sterilization. Now I've already told you a couple ways of moist heat sterilization, boiling in that pot that I just showed you. Autoclave, we've talked about autoclaving. These are using steam, so that it's high heat but with some moisture. Now, moist heat sterilization is oftentimes more effective than that of dry heat sterilization, only because that liquid allows the heat to penetrate the cells a lot better. So let me talk a little bit about pasteurization. You must have heard of pasteurization. It was designed by Louis Pasteur, hence it has his name. And there's basically two types of pasteurization, and you can see these effects on the food in the grocery stores. So most often you'll find your milk in the refrigerated section. All milk that you find at the grocery store has to be pasteurized. So this milk has undergone high temperature, short time pasteurization. So they'll raise the heat to a certain point, at a point that the milk in this case doesn't lose its palatability, so it still tastes good. But it's going to do this for a short time, because if it does it any longer, you'll kill more microbes. Excuse me. But over time, it will actually harm the flavor. So you want to achieve a certain temperature before you harm that flavor and to kill most of the pathogens that are present in that milk. So to do that, it requires a very short time, but because you did not remove or kill all the microbes that are present in that milk now has to be refrigerated. Because it's not sterile, there's still some hopefully non-pathogens present in the milk. Compare the high temperature short time pasteurization with the ultra high. So that means you're going to flash it with these food products with even higher temperature. What this does is it's going to kill more organisms, and it's going to allow you to keep the food product on the shelf for a longer time. So the shelf life is a lot longer, and the food product does not require pasteurization. So I remember the first time I saw, I forget what they call it, it's like parlette. It's this milk that's found in these cartons that's found on the shelf instead of the refrigerator. The first time I saw this, I thought it was not milk, it was something else besides the milk product. But later I learned that there was a new pasteurization technique, and that's this ultra high temperature pasteurization. Okay, this is all the moist heat pasteurization. The other moist heat was that of autoclave, which I mentioned earlier. These are used, you would not pasteurize milk with this because you would have to, this basically allows you to raise the temperatures even higher than pasteurization. If I recall, pasteurization is around 96 degrees Celsius, so it's below boiling temperature, whereas in autoclave you want to reach 121 degrees Celsius. So the temperature is raised way above the boiling point of water. It's used to sterilize items such as surgical equipment. They're designed to kill vegetative cells, viruses, especially endospores. Pasteurization will not kill endospores. It basically it's only used for items that it will not damage. Here's an example of somebody preparing these instruments to be placed in the autoclave. And these would be autoclaved instruments. So these instruments are safe. So first you would wash them, such as the picture on the right. You would then place them in a container and wrap that container. And then you would place it in the autoclave for 30 minutes at 21 degrees Celsius. Now when you take them out, the wrapper outside is not sterile, but all the instruments inside should remain sterile. So here's a picture again, or a slide that compares high heat temperature or heat autoclaving sterilization versus pasteurization. So autoclaves, moist heat, used to sterilize surgical equipment. Pasteurization will kill microbes or pathogens. in particular, and will reduce the total number of spoiling-causing microbes. But it will not kill all the microbes. But the point for pasteurization is to kill those that are potential pathogens. And again, if you were to autoclave milk, not pasteurized, but if you were to autoclave milk, you would not maintain the food quality. It would really be disgusting. So pasteurization allows you to kill the majority of microbes, not reach sterilization, but keep food quality. And again, you know that there's high temperature and ultra high temperature. Okay, that's how temperature affects growth, but you can use it in a way into your benefit to kill microbes. But you can also use temperature at the other extreme. That's low temperature. These will not kill microbes, but they will prevent them from growing. Again, a lot of times infection is due to numbers. If you increase the numbers of a microbe, the more chances of getting a disease and making that disease more severe. So low temperatures, refrigeration, and freezing. So low temperatures are effective at controlling the growth. It will not kill these microbes, but it will prevent them or slow them from growing. Your home refrigerators. Your refrigerated home should be between 0 and 7 degrees Celsius. If it's near 0, chances are you'll get freezing. So your refrigerator at home should be higher than 0, but lower than 7 degrees Celsius. And your freezer at home is roughly minus 20 degrees Celsius. That's why your ice cubes are made in the freezer, because water freezes. This will stop microbes from growing, and it will even kill some susceptible microbes. But generally, if it does kill them, it's because the water crystals have poked a hole through the cell membrane and disrupted the cells. But the majority of microbes will not be killed by that low temperature. What I forgot to mention is your refrigerator, again, it will just slow the growth because metabolism will just happen a lot slower. So these are temperatures that you should be aware of. Your home refrigerator is typically set at 4 degrees Celsius, and your freezers at home is generally about minus 20 degrees Celsius. Now in the labs, we also have freezers that are minus 20 degrees Celsius. We have some that we set at minus 80. Now, when we want to maintain bacterial cultures for a long time in the lab, instead of purchasing them or risking the chance of losing them, we will make a stock culture and we'll stick them in a refrigerator or a freezer, such as the one here in the top right. It is an upright minus 80 degrees Celsius freezer, and we can maintain our cultures for many, many years. As we want them, we take a little bit out, we place them on warm media, and they will continue to grow. Other freezers, like in the lower right-hand corner, this is liquid nitrogen that is liquid at minus 196 degrees Celsius. Microbes will stay there a lot longer time, for forever basically. Tissue culture cells, they tend to be placed in a minus 196 degree freezer. So under the proper freezing media, and at these temperatures, microbes will not die. They will survive. They just will be arrested in their growth until we take them out. Okay, so I've mentioned temperature, and I've mentioned a little bit of pressure, such as the autoclave. Autoclave takes the combination of steam, heat, and adding pressure so that the water boiling can boil above 100 degrees. Otherwise, without pressure, you would never reach 121 degrees. So this concept was also used in the food industry, again, to render bacteria harmless, to preserve the food quality, and if there's less microbes present on the food, that food will stay good longer, so it'll have a longer shelf life. So it's a method of preserving and sterilizing food. You probably did some canning. Canning uses pressure also. So the application is sufficient to kill vegetative cells by denaturing their proteins, but the endospores will still be present. So this slide basically compares heat, whether it's moist or dry heat, versus pressure. Now we know that heat will basically, the important part is that bacterial spores are killed, microbial enzymes are completely destroyed, and it's better for low acid food products. Now pressure, on the other hand, which you should know, and I don't see it here, but it will not kill the endospores. but it's used in its place for food processing because it maintains the taste better of the food product. So you could control the microbial growth of food by either heat or pressure, and you'll choose the one that basically renders the food product more marketable. Okay, besides heat and pressure or low temperature, there's also radiation. And I briefly mentioned the UV ultraviolet light for disinfecting a operating room. So radiation in various forms, from high energy radiation to sunlight, can be used to kill microbes or inhibit their growth. In particular, most people use a form of X-rays and gamma rays because they easily penetrate through plastic and paper. So in the top left picture, actually in all the pictures, I'm showing you some surgical equipment that's being placed in a paper plastic type of container, or I should say wrapper. This wrapper, some of them can be placed in the autoclave, but if you're going to use gamma rays or X-rays, you would not need an autoclave. So you could sterilize, for instance, plastic surgical equipment or plastic medical equipment using gamma rays and X-rays. And you'd wrap them in similar wrappings that I'm showing you here, except you'd zap them with, say, UV light instead. And whatever is inside the package, if irradiated correctly, would stay sterile until that package is open. So besides surgical or medical equipment, food is likewise irradiated. So it's a technique that improves the safety and extends the shelf life of food. It eliminates microbes and insects. Like pasteurizing milk and canned foods, irradiation can make food that much safer. Now, when people hear that their food may be irradiated, they sometimes feel that the food might have radiation and that they might acquire cancer by this. But basically what irradiation is doing is especially if it's UV, it's destroying the DNA that's found in the microbes, but it will not affect your DNA. So when you ingest these foods, you have the right metabolic pathways that destroy or that break down proteins into amino acids, DNA into nucleic acids, etc. So your body will digest these foods the same way with no harm because of the irradiation process. Now, the USDA the USDA has put out some safety guidelines. It's been approved since the year 2000. They do use gamma irradiation. They can use some X-rays. They break down the chemical bonds of the DNA of the bacteria. They do that by allowing the rays to essentially pass through the food, and generally they'll only do it on the surface because, again, that's the part that would be exposed to microbes. It does increase the shelf life of your food because there are little microbes present, live microbes present. And it's marked by this Radora logo showed here on the right. And so the idea is that you'd go to the grocery store and you would see this this type of logo on the food. And I think since this slide that logo has been modified. So I don't think it's the same thing. Okay, so that ends basically sections 13.2. And we'll finish this lecture with section 13.3, in which we will use chemicals to control microorganisms. Okay, your textbook goes through a lot more of these chemicals. I'm only going to go through a handful. Again, these are common household items or items that I believe that you've come across during your everyday interaction. The first ones are halogens. Now, halogens, you probably know it more as chlorines. So chlorines or sodium hypochlorite, you know of chlorine or Clorox bleach. Okay, so you're going to find these chemicals in chloride bleach. So these halogens are chlorine. It could be fluoride. We have fluoride to rinse our mouth. We have iodine to swab the surface skins of patients before undergoing surgery. We use chlorine to keep our pools clean from algae. So these are items that at one point of your life you've come across. So I'm showing you Clorox. Here is my chlorine salt with the sodium, and this is my chlorine dioxide. There's two oxides. So these chlorine components includes sodium hypochlorite, chloramines, and chlorine dioxide. And they're basically used for water disinfection. Those are the chlorines, and then the fluorides are the iodines. I don't have an example for fluorines. The iodines will be both a tincture of iodine or iodophorus. Here's an example of betadine. This is a povidone iodine solution, and this is used to swab the surface of your skin. So in either case, betadine is used as a hand scrub by medical personnel before surgery, and it's also used as a topical antiseptic. So remember, this is going to be used on tissue, so it has to be safe for tissue. So it's not a disinfectant, it's an antiseptic. How it works is that it's going to oxidize cellular components, and it's going to disrupt the nucleotides, fatty acids, and just basically destabilizing them. There we go. The next category I'd like you to know are these phenolics. They have a phenol ring, so I brought these chemical compounds here to show you. Whenever you see a ring like this is called a phenol ring. They are very stable. They last long. They have long-acting disinfectants, and they denature proteins and disrupt membranes. So I should say deactivating antiseptics or long-acting antiseptics, because we're going to use this on tissue. They're commonly found in household cleaners. So if they're in household cleaners, then yes, they can be disinfectants. But if they're used in mouthwash, then they would be an antiseptic. Hospital disinfectants, and are also used to preserve harvested crops. So here's a picture of Listerine Fresh Burst that I found in my house, and listed is methyl salicylic and thymol, and as well as eucalyptol and menthol. Now the thymol and the eucalyptol are both phenolics, and they are the active ingredients in mouthwash. So they're going to inhibit microbial growth by basically denaturing the proteins from the microbes that are present in your mouth. So it's important that you gargle for a minute so that it reaches every corner of your mouth, and so that if there are any microbes left after brushing your teeth, that you further kill those microbes. And so it's that one extra step. Here's a picture of the Listerine Fresh Burst. I also found them in the mint strips. I always wondered if mint strips did the same or if they just freshened your breath. If you look at the label, and as long as it says thymol and eucalyptol, they are antibacterial. They will kill germs, as it says, that cause bad breath, plaque, and gum disease, gingivitis. So besides bad breath, which is very important, but equally important is plaque and gum disease. Alcohols. A lot of you guys may have Purell that you carry around. It'll do the same as rubbing alcohol that's in your cabinet. It rapidly denatures proteins. It disrupts their cell membrane. Again, remember that 95% ethanol in your gram stains. It disrupted the phospholipid membranes. Well, that's what's happening here with Purell or the rubbing alcohol in your cabinet. So they are used as antiseptics because they're safe to use on tissue. They tend to be bactericidal or fungicidal. They may also be virucidal. It might kill the envelopes that some viruses will have. Their active ingredient is instant. And again, you have to make sure that you put enough so that it doesn't evaporate too fast, so that it kills enough microbes before it evaporates. You have a group of chemicals called surfactants. These are surface active agents. They act by basically lowering the surface tension of water. So if you look at the molecule in the upper right hand corner, it's a long hydrocarbon chain. So you know that that's very polar, so it's going to break the hydrogen bonds of water. It's going to disrupt the surface tension, so it's going to spread water. It's one of the major ingredients in all soaps and detergents. So it's not an outright antimicrobial, but it's going to be a surfactant. It's going to help release the microbes and wash the microbes away from the surface. So they interact with nonpolar oils and grease They create an emulsion in water. They loosen and lift away dirt and any microbes that might be attached to those surfaces. So regular soap, not antibacterial soap, but regular soap will have surfactants. And if you wash your hands in the correct way, you will reduce the amount of microbes on your hands, but you will not completely sterilize your hands. So that it's not an antiseptic or a disinfectant. The reason being is because it does not kill microbes. Nonetheless, regular soap is very effective at preventing the spread of microbes. Okay, lastly are peroxides or peroxygens. One of them is hydrogen peroxide. If you have contacts, you will put your contacts in a hydrogen peroxide solution. So they are oxidizing agents. And so remember, as an example would be hydrogen peroxide. And again, you could put that on your skin. It's an antiseptic because it's safe enough to put on your skin. Okay, your textbook mentions a few others, but I'm going to stop there. That's enough information. That will conclude this lecture on Chapter 13.